The CRISPR revolution just went into overdrive. Imagine a technology that could erase antibiotic resistance, rewrite your RNA on the fly, and identify hidden causes of brain disorders and all in the span of 2 weeks. That's not science fiction. That's February 2026. CRISPR has just hit a new gear. We're talking gene drives that hunt down drug-resistant bacteria like guided missiles, RNA editors that bypass DNA entirely, and breakthrough discoveries connecting mysterious genes to conditions like microcephaly and autism. Today, I'm breaking down the most significant CRISPR developments, stuff that's already being tested in labs and could reshape medicine and agriculture within years, not decades. And I promise you, some of this will genuinely blow your mind. Let's get into it. Quick context if you're not deep in the CRISPR weeds. Since 2012, this molecular scissor technology has evolved from a bacterial immune system curiosity to a full-blown genetic Swiss Army knife. The core concept? Precisely cut DNA at targeted locations, then let the cell repair itself ideally with your desired edits. But here's the thing, CRISPR isn't one tool anymore. It's an entire toolbox. You've got base editors that swap single letters of genetic code without cutting, prime editors that rewrite sequences like a word processor, and now, as we'll see, systems that don't touch DNA at all. The stakes are massive. The World Health Organization projects 10 million deaths annually from antibiotic-resistant infections by 2050 if we don't act. Meanwhile, about 15% of genetic diseases stem from splicing errors, problems that traditional CRISPR struggles to address without risky permanent DNA changes. That's the landscape. Now let's talk about what just dropped. First up, researchers at Hebrew University just identified a gene called PEDS1, and it's a game changer for understanding neurodevelopmental disorders. Here's what they did. They took a genome-wide CRISPR knockout screen, basically. They systematically disabled nearly 20,000 genes in human stem cells to see which ones are actually critical for turning those stem cells into functioning neurons. Out of thousands of candidates, 331 genes proved essential. But PEDS1 stood out. When they knocked out PEDS1, something dramatic happened. The cells couldn't form neurons. Brain organoids, those mini-brain models grown in dishes, shrank by up to 50%. That's a massive red flag pointing to conditions like microcephaly, where children are born with abnormally small heads. Then came the validation. They found 2 unrelated families with kids suffering from severe intellectual disabilities and developmental delays. Genetic sequencing revealed mutations in PEDS1. These children had head circumferences averaging 3.5 standard deviations below normal, a clinical hallmark of microcephaly. But here's why this matters beyond one rare condition. PEDS1 likely plays a role in a spectrum of neurodevelopmental issues, potentially including autism. For years, many of these disorders have been genetic mysteries. Families get diagnoses like unspecified developmental delay with no molecular explanation. PEDS1 gives doctors a concrete target to sequence for, and more importantly, it opens pathways for understanding how brain development can go wrong at the cellular level. The limitations, so far we're talking about 2 families and lab models, but this is exactly how medical breakthroughs begin: one gene, one pathway, one clue at a time. Now for something that might sound subtle but is actually revolutionary, editing RNA instead of DNA. Traditional CRISPR cuts DNA, that's permanent, it's in every cell that inherits that edit. For some diseases, that's perfect, but for others, especially conditions caused by splicing errors, where genes are stitched together incorrectly at the RNA level, you need a different approach. And a RESPLICE, announced by the ARC Institute just days ago. And this is a dual CRISPR system using 2 Cas13 proteins, variants specifically designed for RNA, not DNA. Here's the elegance. One protein delivers a donor exon, a chunk of genetic instructions to the target RNA transcript through precise base pairing. The second protein, a deactivated Cas13, blocks the cell's natural splicing machinery from doing its usual job. Result? The cell incorporates your donor exon instead of the problematic native one. In their HEK293 cell experiments, they achieved up to 40% efficiency inserting corrective exons into DMD, the gene responsible for Duchenne muscular dystrophy, a devastating muscle-wasting disease. And they did this without cutting DNA even once. Why does this matter? 3 reasons. 1, it's reversible. RNA edits are temporary because RNA degrades naturally. If something goes wrong, you stop treatment and the effect fades. With DNA edits, you're committed. 2, it tackles a huge disease category. About 15% of genetic disorders involve splicing defects conditions where the right gene exists, but it's assembled wrong. RESPLICE could address these directly. 3, off-target effects appear minimal. Their RNA sequencing showed less than 1% unintended edits elsewhere in the transcriptome. That's a major safety advantage. The catch? This has only been tested in cell cultures. Delivering these complex two-protein systems into human tissues, especially hard-to-reach organs like the brain or muscles, remains an enormous engineering challenge. But if they crack delivery, we're looking at a whole new class of genetic medicine. Think of it this way. Traditional CRISPR is like rewriting the master blueprint of a building. RESPLICE is like editing the work orders that go out to construction crews, still powerful, but with an undo button. Alright, here's where it gets wild. UC San Diego just engineered what I can only describe as a genetic assassin for antibiotic-resistant bacteria. The system is called pPro-MobV, and it's essentially a CRISPR-powered gene drive designed to spread through bacterial populations like a benevolent virus. Here's the problem it's solving. Antibiotic resistance is spreading because bacteria share resistance genes on little circular DNA molecules called plasmids. When bacteria form biofilms, those slimy protective fortresses you find in chronic infections like UTIs or on medical implants, they become nearly impossible to kill with conventional drugs. So the researchers did something audacious. They packaged CRISPR-Cas9 into a conjugative plasmid one that can transfer itself between bacterial cells through natural mating processes. But instead of carrying resistance genes, this plasmid hunts them down and destroys them. In their experiments with E. coli biofilms, pPro-MobV spread to 90% of bacterial cells within 48 hours. It specifically targeted plasmids carrying BLA-TEM1, a gene that confers resistance to beta-lactam antibiotics like penicillin. Once it found them, it cut them apart with CRISPR-Cas9. Result? A 99% reduction in viable drug-resistant colonies. Bacteria that had been untouchable were suddenly vulnerable to antibiotics again. They even tested delivery via bacteriophages, viruses that infect bacteria, and still achieved 70% penetration of the biofilm. And they built in a kill switch, a homology arm that prevents the system from spreading indefinitely into the environment. Let me put this in perspective. By 2050, we're projected to lose 10 million lives per year to antimicrobial resistance more than cancer. This approach doesn't just kill resistant bacteria, it actively reverses resistance in populations, potentially cutting environmental transmission in half, according to lead researcher Ethan Beer. Trials are planned for aquafarms and hospital settings. The risks? Horizontal gene transfer to non-target species is a concern, and will need rigorous ecological monitoring. But conceptually, this is next level, using bacteria's own sharing mechanisms against the very traits that make them dangerous. Quick pivot to agriculture, because CRISPR isn't just for medicine. Cornell researchers just published findings on grapevine powdery mildew resistance, a fungal disease costing the U.S. wine industry $3.7 billion annually. They used multiplex CRISPR to knock out susceptibility genes called MLOs. Specifically, knocking out VVML07 alone conferred 85% resistance. Triple knockouts hit 95% but caused stunted growth. So the sweet spot seems to be selective editing. Under field conditions, edited grapevines yielded 20% more berries, with dramatically reduced fungicide needs. This matters in an era of tightening pesticide regulations and climate stress on crops. Meanwhile, Japanese researchers created cherry tomatoes with a popcorn-like aroma by knocking out TBG1 and Tomlock-C genes. The result? A threefold increase in hexyl butanoate, that buttery, sweet scent without sacrificing yield or nutrition. Same lycopene, same sugars, just better sensory experience. These aren't gimmicks. Aroma compounds drive consumer preference and premium pricing. Disease resistance determines whether crops survive increasingly volatile weather. CRISPR is becoming a mainstream agricultural tool, not a laboratory curiosity. Now we need to talk about the elephant in the room, regulation and ethics. On January 26th, the FDA announced an accelerated approval pathway for one-time CRISPR cures. Companies like CRISPR Therapeutics are celebrating because it fast-tracks therapies through surrogate endpoints rather than waiting years for full clinical outcomes. But ethicists are sounding alarms. Kiran Musnuru and others point to a case known as Baby KJ, an infant who received off-label embryo editing for hypertrophic cardiomyopathy, in 2025 without long-term safety data. The concern that speedy approvals could greenlight unproven edits in vulnerable patients, especially children who can't consent. FDA advisor Marty Makary counters that speed saves lives and and post-market surveillance can catch problems. That's the tension, innovation versus caution. There's also an equity problem. About 80% of CRISPR clinical trials are in high-income countries, according to WHO data. The benefits are accruing to those who can afford them, while low-income regions face the same disease burdens without access to cutting-edge therapies. Nature's January 21st editorial ranked CRISPR among 2026's top technologies, but urged global regulatory harmonization to prevent a designer baby slippery slope. We're at an inflection point where the science is outpacing the governance frameworks. I don't have clean answers here, but it's worth stating clearly. Every breakthrough we've discussed today comes with responsibility. The question isn't just, can we do this? But should we? Under what circumstances? And who gets to decide? So where does this leave us? In 2 weeks, we've seen CRISPR expand from DNA editor to RNA rewriter, from single-cell therapy to population-level gene drives, from disease treatment to agricultural enhancement. These aren't isolated advances, they're building blocks. Imagine 5 years from now, RESPLICE-style RNA therapies treating muscular dystrophy with temporary, tunable interventions. Gene drives deployed in controlled environments to reverse antibiotic resistance in hospital wards. PEDS1. Screening becoming standard for unexplained developmental delays, giving families answers and researchers targets for intervention. The challenges are real. Delivery mechanisms need work. Getting complex CRISPR systems into the right cells at the right time remains hard. Off-target effects need continuous monitoring even with low error rates. And regulatory frameworks worldwide need to catch up to a technology moving at breakneck speed. But the trajectory is clear. CRISPR is transitioning from experimental tool to clinical and agricultural reality. We're not talking decades anymore. Some of these applications, particularly the antibiotic resistance work and agricultural edits, could see commercial deployment within 2 to 3 years. That's it for today. If this kind of deep dive tech analysis is your thing, subscribe and hit the bell. I'll be tracking CRISPR and other transformative technologies as they develop. Drop a comment with your biggest question or concern about gene editing. The future is built by Science Bids. Thanks for watching, and I'll see you in the next one.